So in this second part of the talk, I'm going to highlight how some of the tumors that you've seen in the first part of the talk in cell cutaneous syndrome have been studied using novel genetic techniques. The learning outcomes for this part are that we can use genetics to help delineate what might be underlying a range of clinical and histological phenotypes of tumors that are arising in a rare genetic condition, and that sometimes the analysis of this highlights mechanisms that might be relevant for the development of new treatments. In addition, we show how that different mutations can be associated with different tumor phenotypes. To recap briefly, this is cell cutaneous syndrome, and these are a range of tumors that arise in these patients, which are often clinically labeled as benign. But we haven't really gone into the pathology of these tumors, and I think that's important to make clearer as to why clinically the appearances are different. So cylindroma and spiradenoma are two different types of tumors, which are clinically different in terms of symptomology. So cylindroma is often painless, spiradenoma is painful, but histologically, cylindroma is a highly organized tumor with tumor cells arranged in nice, neat circles. And in spiradenoma, there is a loss of this organization, and there are also many tumor-infiltrating lymphocytes. And in 1840, Bill Roth had seen these circles on a biopsy specimen at the time, and that led to the coining of the term cylindroma. Now, interestingly, you can sometimes get the two tumor patterns in the same excision sample, and in the past, this was considered to be a collision event between two different tumors, but we've gone on to show over time that actually it's really the same tumor. However, within that tumor, there can be different levels of organization, with cylindroma at the organized spectrum, and intermediate, which is cylindrospiradenoma, and finally a disorganized version, which is called spiradenoma alone. And this spectrum of tumor phenotypes, it doesn't simply restrict to the benign tumors. We think in these patients, you can also get more clinically malignant patterns, and also some patterns which are clinically and pathologically malignant. And I'll go over some of those now. So there are tumors that can present which have a malignant clinical course, but when the pathologist looks at the sample that's been biopsied, it's falsely reassuring. So this is an example of a patient who's had a cylindroma where there's been local invasion through the skull plate into the brain, but the histology is reassuring. Separately, we've had a patient in Newcastle where there was a 20-year history of smoking and a new presentation of breathlessness, and imaging revealed multiple lesions that were sat within the lung parenchyma. And when we did FDG PET on these, we showed them to be active and they lit up in yellow, as seen in the middle panel there. And when we did bronchoscopy, we showed evidence of tumors that were blocking the large airways and interestingly had arborizing blood vessels over the top, much like you would see in cutaneous cylindroma. Again, the histology that we had from the biopsies was very reassuring. Sarah Fennick, who I mentioned earlier, went on to highlight these tumors in the skin in green on the panel on the left, and the pulmonary tumors on the panel on the right in yellow. And when she compared the tumor kinetics in the skin and in the lung, she found that the lung tumors were growing more quickly. These patients can also develop salivary gland tumors in a small proportion, and these are typically of a low-grade tumor type. Now, in addition to those tumors, patients can get malignant tumors, which are really quite rare, but are adenocarcinomas in the skin, and they can be of different histological types. In addition, sarcomatoid neoplasms can be seen. There are also presentations of a special subtype of squamous cell carcinoma called follicular squamous cell carcinoma, which can arise in these patients. In fact, there have been a range of cases of malignant cylindroma and spiradenoma, which have presented from as early as early 40s through to 78 in the literature, where there's metastases to other tissues such as lymph nodes, the liver, and bone. And this very first case was reported from a patient in Birmingham in 1842, and that was a young person who presented with metastatic disease to the abdomen. So to help better understand what was happening in terms of the genetic changes in these range of histological patterns of tumors, as well as clinically variable malignant behavior, we wanted to do whole genome sequencing of a range of tumors from 2 severely affected individuals in our large family. And when we did this, we did it in collaboration with Helen Davies and Serena Nick-Zainal at the Sanger at the time. And we looked to see what may be present at the whole genome scale. And we found that there was loss of heterozygosity at 16q as the key event. In addition, we did whole exome sequencing on an expanded number of cases, such that we had data from 13 patients and 5 families. And across these tumors, it was very striking that there were very few mutations per megabase, which is exceptional for a skin tumor. And in fact, only a minority of tumors, which were labeled as malignant clinically, had more mutations. This is a chart that has on the top row a key of the different histological patterns of tumor presentation, and then in the subsequent rows, information on mutations in genes which have been studied in these tumors. And so in the first row, we see that the majority of tumors that we've studied are the cylindromas and spiradenomas, which are the main part of the disease in CCS. But we've also included some basal cell carcinoma, some pulmonary cylindromas, and some malignant cylindrocarcinomas. And importantly, across almost all of the tumors, we see biallelic inactivation of SILD, either through loss of heterozygosity or a second new mutation in the remaining normal allele. So the 2-hit hypothesis and the tumor suppressor gene story does hold with this. But the new and interesting thing here was that when we found, when we studied these tumors, was that we found additional mutations in epigenetic modifiers. And so these are genes that encode proteins that do not change the sequence of another piece of DNA, but alter the level of gene expression of those associated targets, if you like. And so these genes normally play a role in maintaining levels of gene expression. And when they're mutated, they appear to be selected for in these tumors, which is why we detect them in these studies. Now, interestingly, because we had multiple samples from a patient, we could study the mutations in different tumors, and we found that tumors had their own specific mutations in these genes. Sometimes these mutations were there alone, sometimes they were there in tandem. And so we were quite convinced that we were seeing a pattern that wasn't due to a single clone spreading to different tumors, because the location of these mutations were at different sites. In addition, we did a validation technique called deep sequencing, where we did very focused next generation sequencing of certain genes that we were interested in, and found that in a range of tumors, these mutations were present, sometimes at smaller and smaller levels, suggesting that within a sample, subclones of these tumors may exist. We found that these mutations largely were in the methyltransferase domain, but interestingly, many of them were restricted to the short isoform of this enzyme. When we looked at different levels within a tumor, because if you have a large gray tumor as indicated here, you can section through it and take DNA from different parts of it, we found that some mutations were present in all of the tumor, whereas some mutations were only present in certain levels of the tumor, suggesting that there were subclones again within a single tumor. We even went as far as to microdissect out regions of a single section and demonstrated here that with different levels of organization, levels of these different mutant allele frequencies varied. So this seemed to point the finger at DNMT3A a little bit and BCOR. And in DNMT3A, we studied the gene expression using a separate technique called RNA sequencing. And when we did this, we found that there was an overexpression of the short isoform of this in all of the tumors. And so it seems to be that once you lose both copies of the SIL gene, this isoform is preferentially alternatively spliced and expressed. And then because the function of this isoform is to be involved in methylation, we did methylation profiling of some of these tumors. And we found that if you had a mutation in DNMT3A2, there was a different methylation profile compared to the situation where you didn't have this. And when we looked to see what the most variably methylated genes were in the mutated state, these were actually Wnt signaling pathway genes. We also looked at RNA sequencing data from matched RNA from these samples and found that the RNA sequence demonstrated a different Wnt target gene profile in the DNMT3A2 mutated tumors. Now, this was interesting because in mice, when they've done knockout models of DNMT3A, there is an increased predisposition to skin tumor formation, and these skin tumors appeared to have altered Wnt signaling. So to make sense of this, we looked at how all this might tie together. And so if you imagine right at the beginning, cylindromas are thought to arise from hair follicle stem cells. And this is a single cell in a hair follicle of a patient with Syl cutaneous syndrome. One copy is normal of the Syl gene, the other copy is the mutated tumor suppressor gene. We then have a scenario where a second hit happens, and this results in both copies of the Syl gene being inactivated, and we feel that this is enough to form a benign cylindroma. When this happens, we know that Syl normally negatively regulates Wnt signaling through a separate mechanism, and this results now in a loss of this break on Wnt signaling and an increase in Wnt signaling in tumor cells. We feel that a response to this might be an alternate expression of DNMT3A2 to act to try to control this. However, when DNMT3A2 is mutated, this increases the Wnt signaling further, and this means that these cells are given an additional survival advantage, which is why we're now detecting them in our sequencing data. And this is interesting because this is a second point in the literature where we have found Wnt signaling as an attractive target in these tumors. And as we're going forward, molecules that are being developed in other cancers, such as colorectal cancer, could be applied in these tumors. And we are testing some of these in tumor spheroids. Now, I mentioned the lung tumors previously, and these tumors had a mutation in a gene called AKT1. Now, the particular mutation they had was not present in any of the skin tumors, and also was a treatable mutation. And so this is something which we're currently pursuing by trying to treat our patients with pulmonary cylindromas with AKT1 inhibitors. In one of the malignant cylindrocarcinomas, we found a range of different mutations which were not present in any of the other tumors. One of these were in a gene called MBD4, methyl binding domain protein 4. Now, this was an interesting mutation to find because in this family, everyone in that family had a heterozygous mutation for this. But in this malignant cylindroma, there was a homozygous mutation present. And so it appears that biallelic mutations in MBD4 are present in malignant cylindrocarcinoma. In addition, we also found a different malignant tumor had inactivation of p53 by mutation, as well as a further epigenetic modifier, EP300. So the malignant tumors that arise in these patients histologically appear differently, but that's also something that correlates with mutational status. And Akhtar Hussain, the pathologist that was key to helping us phenotype a lot of these tumors, is indicated in the top right. Finally, we had a basal cell carcinoma in one of our patients, and interestingly, this showed both SILD and patch1 mutations, and patch1 is a recognized driver in BCC. Now, one of our patients had two tumors which were next to each other and presented at the same time, and one of them was a trichoepithelioma with very few mutations per megabase, and the other was a BCC, which had the highest number of mutations in our series. And this made us think about whether these tumors might be arising from different stem cell compartments in the skin. And we think one of the explanations as to why we've got so few mutations in a skin tumor in our condition is that unlike many of the other skin cancers which have been sequenced where there are many, many mutations, the cells that are key for starting tumorigenesis are deeper in the skin in Syl cutaneous syndrome and may not be as susceptible to UV-induced mutagenesis. Now, with Serena's lab, Z was really important in helping us compare mutations between tumors at sun-exposed sites and sun-protected sites, and to do mutation signature analysis. And attractively, when we did this, we found that sun-exposed sites had a UV signature, signature 7, shown here in yellow, whereas sun-protected lesions who had the same histology, did not have this UV signature. And so we highlight that in this condition, that there may be two methods to tumorigenesis. There is also a scenario which we were unable to explain before that mutational signature analysis helped with. And this was the pulmonary cylindromas. We didn't know whether this was because of smoking or whether this was actually metastases from the skin to the lung. And attractively, when we studied these lung lesions, they showed evidence of a UV signature, suggesting that they had originated in the skin and metastasized to the lung. So to bring this all together, we feel that cilled biallelic activation is enough to give you a benign primary cylindroma. But if you have an additional mutation in either DNMT3A or BCOR, which are negative regulators of Wnt amongst other targets, you have increased Wnt signaling, and this gives those clones a survival advantage. And this is a feature of benign cylindromas and spiradenomas. So we've already had 2 hits in our hypothesis, and this could be the 3rd. Now, if you have an AKT1 mutation, you go down the route of having the scenario of pulmonary cylindromas. And finally, if you have mutations in other genes, such as MBD4 or the loss of P53, then you get malignant cancers, which are unique to this condition because they arise in the context of biallelic cilled loss. And so with that, I'd like to summarize that the mutational landscape of these tumors has given us the ability to understand better the heterogeneity in clinical presentations we see, the range of histological patterns that pathologists will see, and also pointed us to new targets that we can think about to take to early phase clinical trials in the clinic. With that, I'd like to highlight all the different funders and colleagues that have supported and helped drive this work, and I'd be happy to take any questions. Thank you.